Hi, my name is Kristin Brogaard, and I'm co-founder and chief scientific officer at Inherent Biosciences. Today, I look forward to talking to you about the discovery of an epigenetic biomarker predictive of sperm quality. Now, the problem we're trying to solve is that we know there are limitations in the standard semen analysis when identifying male factor infertility. Additional technologies are definitely needed and would improve male reproductive health. And specifically, our mission at Inherent Biosciences is to improve the current standard of care by identifying male factors that are missed by the standard semen analysis. This is important because when men are missed by the standard semen analysis, women are subjected to procedures that will not work. Now, to do this, we're utilizing a technology called epigenetics and specifically DNA methylation. Epigenetics is the control of DNA and gene expression that is actually independent of the DNA sequence itself. DNA methylation is one mechanism of epigenetic control, and it's the binding of a CH3 group, the methyl group, to the DNA backbone. In large generalization, we assume that when there is hypomethylation, so when there are few methyl marks on a gene promoter, it actually turns on gene expression down the gene body. When there's hypermethylation, or lots of methylation groups in the gene promoter, it stops gene expression. What's really exciting about this and a potential for future therapeutics is that we know DNA methylation is influenced by the environment. So the environment can turn on and off genes by adding more or less DNA methylation marks. What we did, and was really a new approach in analysis of sperm epigenetics, is we developed this concept of epigenetic stability in sperm. So specifically what this means is when you look at all the genes in fertile or healthy men, here we see gene A and gene B, and line up their promoters. What we consider a stable promoter is when each of those promoters in a healthy or fertile man are consistent. So you can see in this person 1 through 4, these methyl marks are consistent in gene A, where they are inconsistent in gene B. So we would consider gene A having a stable promoter. What we identified in men is that there was 1,233 promoters that were very stable in fertile men. So you can imagine that these promoters had evolutionary pressure to remain very, very regulated and be working so a sperm can be a sperm. What we found was that increased instability. So within these 1,233 promoters, when you start to become unstable, that's associated with a subfertile phenotype. And we saw this on an individual level. So looking at all 1,233 genes, here in a fertile control versus a subfertile individual, the x-axis is all 1,233 genes, and the y-axis is the stability of those genes. We defined cutoffs for what's a healthy stability within those genes, and that's what's seen in the black line. And you can see in the fertile control, there's only a few promoters that go above this line. But in the subfertile individual, there's a large accumulation of instability in these promoters that we've defined as very important for sperm to be a sperm. We also saw this increased accumulation in a subfertile phenotype and dysregulated promoters at a population level. So with a large number of men seeking fertility care versus fertile sperm donors, we saw that there was an increased number of unstable promoters at these 1,233 genes between the two groups with a significant p-value. Utilizing this concept and access to a large multi-site NIH study of 1,311 semen samples, what we were able to identify were 3 groups of accumulation of unstable promoters that were associated with different fertility levels. So we found that there was excellent fertility potential when you had the lowest number of unstable promoters, a normal fertility potential with the next level, and then when you start to get accumulation of too many unstable promoters, that turned into a poor fertility potential. This concept of accumulation of promoter dysregulation and instability is the basis for our test called the sperm quality test, or SpermQT. And that's what I'll be referring to it from now on. We are now marketing SpermQT under the brand Path Fertility. Specifically, what we saw with SpermQT is that individuals undergoing IUI had very distinct pregnancy and IUI and pregnancy and live birth outcomes within these different classes of the SpermQT result. Here we see between excellent, normal, and poor, for pregnancy and live birth rates, we saw a statistically significant difference in number of pregnancies and live births between these groups. And this is men undergoing IUI with an N of 554. What was really neat about this is that when you looked at those men with poor SpermQT result, we looked at which promoters were dysregulated. And what we found was that subfertility within these groups is multifactorial and heterogeneous. So no man had the same combination of promoters being dysregulated. Some promoters, which were really fascinating, occurred more often. This is up to about 35% of the men, but really every single man had a different combination of promoters being dysregulated, which really shows that male infertility is a complex biological condition and should not be treated as the same between all men. There was no correlation identified between the SpermQT results and number of dysregulated promoters with male age, motility, sperm concentration, and DNA fragmentation using the HALO method. Importantly, we saw men with a poor SpermQT result. Of those men with poor SpermQT results, 82.5% of them had normal semen parameters as measured by concentration and total motile count. So these men in the poor group would have been missed by the standard semen analysis and subjected to procedures that will not work. Also importantly, when you combine SpermQT and semen analysis, you get even more predictive pregnancy and live birth rates. So here we have a SpermQT result. The total motile count split between greater than 20 million and less than 20 million, which were the WHO cutoffs, pregnancy rate, and live births. And you can see that when you start to get poorer total motile count and SpermQT result, you get much more granular and predictive pregnancy and live birth rates. Interestingly, we didn't see this same phenomenon with IVF and ICSI, suggesting that ICSI overcomes the sperm quality issues seen with SpermQT, similar to ICSI overcoming issues seen with semen parameters. So here's the graph that you've seen before with IUI. Here are individuals undergoing IVF with ICSI, average 2 cycles, and there's no statistically significant difference between pregnancy and live births between these groups. In the clinical data that we've already collected, the SpermQT prevalence is pretty interesting, where 25% of men within these groups have poor sperm quality, about 11% have excellent, and the remaining have normal. We are very data focused, and we have ongoing studies going with multiple sites, looking at the number of men with poor sperm quality, how it's associated with outcomes. As we are collecting outcomes, we do see that there is a higher number of unstable promoters at clinical sites versus a new group of fertile controls. We also see a shift in the number of unstable promoters of men who are not pregnant versus pregnant from IUI, but our N's are very low. And we are continually looking at the genes that are associated with the poor sperm quality and subfertility. So why is this important? Men with normal and excellent sperm quality results in combination with normal semen parameters can provide, the combination of these two can provide physicians more confidence that there's no male factor infertility. Identification of men with poor SpermQT, you can actually direct treatment much more quickly out of IUI to ICSI to get to a baby faster. And in the future, we're very interested in understanding how DNA methylation is influenced by the environment. This includes lifestyle, behavior, antioxidants, and therapeutics to understand if a man has a poor sperm quality, can he actually reverse that? So what's next with Inherent Biosciences? Again, we are very focused on data, and we are continuing data collection from independently initiated studies to make sure we understand how SpermQT is associated with outcomes, with the goal of validating our current data and expanding our claims. And right now we have 24 nationwide ongoing clinical pilots utilizing SpermQT. And anyone interested in being a part, we would love to have you be a part of our team and help us optimize male factor infertility studies. Thank you.